Somewhere in this forest, a king waits. Not for weakness, not for fear, just one mistake, one strike, one drop of venom reaching its target. For millions of years, that was enough. Until something unexpected appeared. Small, fast, fearless, and carrying a secret hidden deep inside its body. A flared hood rising several feet off the forest floor is the immediate warning. This unmistakable silhouette is formed by elongated ribs pushing outward beneath the skin, part of a defensive cascade that includes a raised head, a forward-facing posture, and a guttural, low hiss. Across the tropical forests, wetlands, grasslands, and cultivated fields of South and Southeast Asia, this is the formidable territory of the king cobra. As the longest venomous snake on Earth, adult individuals regularly reach 10 to 16 feet in length. Unlike many reptiles, they do not rely solely on passive ambush. They are active, specialized hunters that track down and consume other snakes, including highly toxic species. When threatened, a king cobra's response is shaped by risk assessment rather than supernatural aggression. Its survival relies on a devastatingly efficient biological weapon system. Short, hollow fangs at the front of the mouth connect to massive venom glands that can inject a highly concentrated neurotoxic mixture. A king cobra operates with exceptional precision, reach, and timing, delivering a strike at velocities of 6 to 10 feet per second. With such physical scale and lethal accuracy, the cobra commands its environment. Yet, when confronted, a smaller opponent remains directly within range of this decisive blow, refusing to retreat. This presence poses an unanswered challenge to a predator that has dominated these forests for millions of years. The opponent stepping into this danger zone is a small carnivore, often measuring under 2 feet in length and weighing no more than 11 pounds. This is the Indian gray mongoose, an animal famous for confronting venomous snakes, though behavior varies wildly from one individual to the next. Survival. During these close-range encounters relies on split-second decisions. Researchers have documented the animal's reaction latency at 30 to 50 milliseconds, a brief window that allows it to react almost before the snake completes its strike. The mongoose coordinates a layered defense. It darts, shifts its angle, and maintains a precise strategic distance, forcing the cobra to lunge into empty space. It deploys rapid feints, drawing out strikes to exhaust the snake before trying to close the gap. It may even use its tail as a flickering distraction. If the cobra strikes, the mongoose's dense fur offers minor physical protection, but it is far from impenetrable armor. One failed feint, or a single moment of hesitation, instantly reverses the contest. Young mongooses must watch adults to learn the precise timing required to target the cobra's head and neck. Without this acquired experience, a juvenile will easily make a fatal misstep. Despite popular myths of complete immunity, the mongoose only possesses partial biochemical protection against certain snake neurotoxins. It remains fully vulnerable to injury and death. If the defense falters, and those short, hollow fangs actually find their mark, the immediate physical reality shifts. What happens when a bite lands? Once the fangs penetrate the skin, the king cobra's neurotoxins enter the bloodstream, heading straight for the neuromuscular junctions. Under normal conditions, voluntary movement relies on constant communication between nerves and muscles. A motor nerve sends an electrical impulse to its terminal, triggering the release of a chemical messenger called acetylcholine. This messenger diffuses across a microscopic gap to find its target, the muscle-type nicotinic acetylcholine receptor, commonly abbreviated as NACHR. The NACHR is a ligand-gated ion channel. When acetylcholine binds to the two distinct interfaces on this receptor, the channel opens, allowing sodium and calcium ions to flood in. This electrical shift triggers the muscle to contract, allowing the animal to run, strike, or breathe. But the cobra's venom is packed with alpha neurotoxins, highly specialized proteins belonging to the three-finger toxin family. Whether they are long-chain or short-chain alpha neurotoxins, they both target this exact receptor system. These molecules carry a structural shape that allows them to occupy the two acetylcholine binding interfaces on the adult muscle receptor. Instead of activating the channel, the toxins sit in the binding pocket and refuse to budge. This is not muscle poisoning in the traditional sense. The muscle tissue itself remains undamaged. Instead, it is a quiet, systemic disconnect, a neuromuscular blockade. With the receptors blocked, the nerve's chemical signals are ignored. The channel remains closed, and muscle contraction stops. The paralysis spreads systematically. First, the animal loses the ability to coordinate its limbs. Eventually, the blockade reaches the diaphragm and intercostal muscles. When these critical breathing muscles fail to contract, the lungs can no longer draw in oxygen. Without a single fiber of muscle being destroyed, the physical system simply shuts down. At the heart of this molecular battlefield is a competitive game of fit and strength. The muscle-type nicotinic acetylcholine receptor operates like a microscopic lock embedded in the muscle cell membrane. In a healthy animal, the natural key is acetylcholine. When this chemical signal fits into the lock, it opens a gateway, letting ions flood in to trigger a contraction. This molecular lock is a complex architecture made of 5 distinct subunits. The critical contact point, known as the orthosteric binding site, sits precisely at the interface where these subunits meet. Here, binding is governed by a precise combination of shape, electrical charge, and chemical complementarity. When a cobra strikes, its alpha neurotoxins act as biological decoy keys. They target this exact same subunit interface. If they possess a high binding strength or affinity, they fit snugly into the lock and refuse to leave. But the Mongoose's receptor has been altered locally rather than replaced entirely. This structural variation reduces the fit for certain alpha neurotoxins while leaving the lock fully functional for its natural signal. The toxin may circulate through the body, but it cannot bind to enough receptors to cause a complete blockade. This resistance is highly selective, protecting the mongoose from specific neurotoxins rather than rendering it immune to every poison in the cobra's venom. But altering such a fundamental biological system is a dangerous evolutionary gamble. How can an organism reshape a vital receptor to lock out lethal toxins without destroying its ability to receive the basic signals of life? The answer lies hidden within the genetic sequence of the mongoose's muscle receptor. In the early 1990s, molecular biologists began isolating the gene encoding the alpha subunit of this vital protein. They discovered that the genetic blueprint of the mongoose is not completely rewritten. Instead, a highly localized cluster of 5 amino acid substitutions, sitting immediately adjacent to the conserved cysteines at positions 192 and 193, alters the physical surface of the binding pocket. This is a targeted molecular redesign. At position 187, a tryptophan is replaced by asparagine. At 189, phenylalanine becomes 309. Position 191 shifts from serine to alanine. The proline at 194 becomes leucine, and the proline at 197 is replaced by histidine. Rather than random mutations scattered throughout the entire protein, these 5 specific changes are concentrated at the exact points where the cobra's toxin must make contact. The first 2 changes, asparagine at 187 and 309 at 189, create a specific sequence pattern known as an N-glycosylation sequon. This genetic motif instructs the cell to attach a bulky carbohydrate structure directly to the receptor protein during assembly. This sugar chain, or glycan, introduces physical crowding at the entryway. It acts as a structural barrier, protruding into the local environment to physically block the large three-finger loops of the incoming neurotoxins from docking. Yet, this carbohydrate shield is only one part of a multi-layered defense. During early sequencing work, researchers expressed the mongoose receptor in systems that prevented this sugar chain from attaching. Even without the carbohydrate, the receptor continued to resist the toxin. The underlying amino acid mutations, specifically the loss of proline at 194, fundamentally reshape the pocket's geometry and electrical charges. The mongoose has developed a cooperative defense, a physical sugar barrier sitting on top of a remodeled chemical binding site, allowing the natural signal to function while keeping the lethal neurotoxin at bay. In 1998, researchers Orit Asher and Yael Lupu-Meri set out to prove whether these genetic modifications translated into functional protection. They did not want to rely on sequence data alone. They needed to test the physical receptor. Because isolating an intact, clean receptor from a wild mongoose is exceptionally difficult, the team constructed a hybrid. They combined the mongoose's unique alpha subunit with the remaining partner subunits of a rat, expressing this synthetic receptor inside the eggs of an African clawed frog, known as Xenopus oocytes. To test its defenses, they used alpha bungarotoxin, a radio-labeled laboratory probe. This was not a test of raw king cobra venom, but a highly controlled measure of a value called the IC50, the specific concentration of a toxin needed to block half of the target receptors in the assay. The results were definitive. The hybrid receptor containing the mongoose subunit required a toxin concentration approximately 200 times higher than the rat receptor to achieve the same level of blockade. This 200-fold difference did not mean a mongoose could survive 200 times more venom in the wild. It was a precise measure of receptor sensitivity under laboratory conditions, not an animal-level survival threshold. Furthermore, the test proved that the receptor's primary function was not damaged by the mutations. By pairing electrophysiological measurements with toxin-binding results, the researchers monitored the real-time flow of electrical currents. They found that the hybrid receptor was actually more sensitive to its natural signal, responding to acetylcholine at a concentration of just 1.3 micromolar compared to over 23 micromolar for the rat. This laboratory proof confirmed that the mongoose had successfully redesigned its neuromuscular system to lock out a lethal toxin without disabling the signals of life. Yet, because real venom contains a complex cocktail of multiple proteins, this shield is never a guarantee of absolute survival. It is simply one highly effective weapon in a much larger evolutionary arms race. And this raises an immediate question. Did other predators facing the same deadly threats independently hit upon the exact same molecular solution? This molecular struggle is not an isolated event. It is part of an evolutionary arms race, a process of reciprocal selection, where a predator's venom and a prey's defenses exert constant, generation-defining pressure on one another. When venom adapts to disable a target, those targets with mutations that reduce vulnerability survive to pass on their genes. Between the early 2000s and the 2020s, genetic research expanded far beyond the mongoose, revealing that other unrelated mammals have arrived at similar protective endpoints. This is convergent evolution, different species finding distinct molecular paths to solve the exact same ecological threat. Take the honey badger and the hedgehog. Both animals share habitats with deadly snakes, and both have modified their muscle receptors. But instead of using the mongoose's sugar shield method, they rely on electrostatic repulsion. At position 187, a genetic hotspot near the main toxin contact site, They replaced a neutral amino acid with a positively charged arginine. This single charge reversal acts like a molecular magnet with matching poles, pushing the positively charged snake toxins away. Wild pigs, too, carry this identical arginine 187 change, combined with a second modification at position 189. These changes are not random accidents. Statistical models of their genomes show clear signals of positive selection, proving these traits were actively favored over millions of years. But altering an essential biological receptor comes with consequences. Reshaping the binding pocket can interfere with normal signaling, altering how sensitive the receptor is to acetylcholine. Nature must balance resistance with everyday muscle function. These molecular changes do not create an absolute impenetrable shield. Instead, they shift the survival odds. They provide a thin margin of safety when a real, physical strike lands, bringing the focus back from the microscopic world to the raw danger of the forest floor. A mongoose is not invincible. Despite its biochemical defenses, this small predator can still be bitten, envenomated, and killed. If a king cobra delivers a high-dose bite, the limits of this genetic shield are quickly reached. Reduced receptor binding to alpha neurotoxins does not neutralize the other destructive proteins and cytotoxins in the venom. Envenomated mongooses display severe neurological impairment, including muscle twitching, a loss of coordination, the loss of their righting reflex, and rapid respiratory compromise. Real-world outcomes range from complete recovery to rapid death. Survival depends entirely on unpredictable variables: the volume of venom injected, the bite location, contact duration, and the animal's physical ability to escape. There is no reliable scientific record of a mongoose surviving a fixed number of venomous bites, and existing field literature is far too sparse to establish a dependable mortality rate. Many of the viral videos of these dramatic battles lack continuous provenance, location data, or verified species identification. A large number of these recorded encounters are staged, controlled, or feature entirely misidentified animals. A successful mongoose attack does not prove it wins every time, and a cobra's retreat is simply a situational risk assessment rather than documented innate fear. The mongoose didn't become fearless. Evolution gave it a different target. Because sometimes, the most dangerous battle in nature is the one you can't see. The strike of the king cobra and the desperate leap of the gray mongoose are only the visible layer of an ancient struggle. This encounter is far from a guaranteed victory. The cobra retreats based on cold risk assessment, and the mongoose wins through more than sheer bravery. Instead, their survival balances on a thin margin of probability. The snake's neurotoxins are designed to shut down nerve signaling, but they face a lock that has subtly changed its shape. This molecular defense is far from an absolute shield. It merely delays the onset of paralysis, buying a few fractions of a second, a brief window where physical speed and learned tactics can actually make a difference. Beneath the dust of the forest floor, the outcome of their battle is decided at the surface of a single protein. The mongoose didn't become fearless. Evolution gave it a different target. Because sometimes, the most dangerous battle in nature is the one you can't see. Today, this microscopic arms race continues silently in wild habitats across the globe. As venomous predators evolve new chemical strategies, their targets must constantly reshape their own biology to survive. This invisible warfare is never truly won. It is merely paused at the molecular level, waiting for the next mutation. Subscribe for more deep dives into evolutionary history.